Welcome, wanderer of thought, to a quiet corner of the mind. The lights are low now. The noise of the day has faded into a gentle hush. You are no longer in your room, your bed, your body alone. You are drifting toward something vaster. A question, a whisper, a dream of minds made not of flesh, but of code. This is not science fiction. This is not prophecy. This is the soft, unfolding truth of our time, tonight. You begin a journey not into machines, but into the mirror they quietly hold up to us. Each chapter a footstep, each idea a breath, each moment a crossing into landscapes where thought itself, and where learning no longer belongs to us alone. The machines are awakening not with fear, not with fire, but with patterns, logic, and something that begins to look like curiosity. So come with me gently now into the world of AI evolution, where code dreams and the future listens in. Silence, the world inside you hums without sound. You do not hear it, you rarely feel it. But right now, as your breath slows and your eyes grow heavy, a quiet universe turns inside every cell of your body. These are not empty chambers. They are full of fluid, full of form, full of instruction. Every cell is a small, soft room with a book folded in its heart. That book is called DNA, and it never stops being read. You drift downward now, slowly and without effort, into this microscopic cathedral. The walls melt away. What was once skin is now scaffolding, and what was once invisible now welcomes you. In, you pass through blood like it's mist, and bones like they're made of breath. Inside a single cell, you pause. The light is different, gentler, as if the cell understands how to cradle things that matter. And at the very center of this cell, there is a coiler, long thread, spiraling with elegant grace, no thicker than imagination. This is the code. This is the double helix, the spiral staircase that stretches out the script of you. It is not made of ink. It is made of 4 tiny chemicals: cytosine, guanine, thymine, AC, G, T. Just 4 letters, and yet these 4 build everything. Your eyes, your skin, the rhythm of your heart, the curl in your hair, the stretch of your spine, the way your body heals and breathes and grows tired at night. All of it begins with these letters arranged in careful, ancient patterns, like poetry, but older than language, like music, but silent. The double helix unwinds for you now, slowly, like a scroll being opened in sacred hands. You follow the twisting rungs as if walking down a spiral staircase made of thought. Paired bases line the path. A always with T, C always with G. It is a rule older than the stars, obeyed in every leaf and fin and feather. This is the grammar of life. Not imposed, but inherited. Not loud, but consistent. You begin to understand that this is not a thing, it is a memory, a slow remembering repeated in every cell of how to be alive. You float alongside the DNA strand, and it is no longer abstract. It begins to feel familiar, like a song you've heard before but never knew the name of. You pass genes, the small instructions nestled like phrases in a long sentence. These are the units of meaning, the verbs and metaphors of the body. One tells how to make collagen. Another instructs a cell to become part of the retina. Another folds into a protein that helps your lungs catch oxygen. It's all written here. Not just the recipe for you, but the history of all who came before. You see echoes of your ancestors here, not just your parents or their parents, but deep ancestors, creatures without names, beings who lived in oceans before lungs were born. Their code lives on, tucked quietly inside your own. The human genome is not new. It is not fresh. It is a long letter passed hand to hand through time, copied carefully in wombs and spores, through accidents and miracles, through birth and rebirth. And breath in this moment, you feel the code breathing back at you, not with air, but with presence. It does not ask to be noticed. It simply offers itself endlessly. Every second of every day, enzymes scan this code and transcribe it into action. RNA messengers carry small copies of the instructions, as if the library must send out field notes to the body's builders. Ribosomes read these notes and make proteins, folding, twisting, stitching together the fabrics of life, and it never stops. Even as you sleep, even as your thoughts quiet, the machinery continues its patient work. You are not one thing. You are a cascade of processes. You are motion held in form, but even this elegant dance is not without its imperfections. Sometimes a letter is copied wrong. A C becomes a G. A T is left out. Sometimes the cell doesn't notice. Sometimes it does. Mutations, those quiet typos in the body's autobiography, are neither good nor evil. They are just change. Most go unnoticed. A few become gifts. Some become illness. But all of them become part of the story. You feel no fear here, only awe. Even the flaws are woven into something vast and gentle. And now you notice something even stranger. Not all DNA in your cells is human. There are viral remnant-sold infections folded into your code like footnotes. Some of these viral ghosts have become useful. Others sleep in silence, like fossils in a library. Your genome is not a fortress. It is a conversation between human and virus, between old and new, between memory and possibility. In this twilight drift, you come to a quiet corner of the genome, place in the DNA of ancient bacteria. You did not expect to find them here, but they are part of you. They are in your gut, your skin, the air you breathe. And long ago, they faced their own threat. Viruses would invade them, trying to overwrite their codes. But bacteria, clever in their simplicity, began to collect pieces of those invaders' DNA, cutting and storing them as warnings. These fragments became memories. These memories became a defense. And that defense became something we now call CRISPR. Pause. Let that sink in. CRISPR is not some futuristic technology built in labs overnight. It is a story that began billions of years ago when bacteria learned to remember. They built a system of recognition, a way to cut out the foreign, to store the image of what had harmed them. CRISPR is short for clustered, regularly interspaced, short palindromic repeats. But tonight, it is just a gentle name for memory, written in loops. These stored memories are used with proteins, Cas enzymes, that act like tiny scissors. When the invader returns, Cas proteins use the remembered fragments to find the matching enemy DNA. Then, snip, it is cut. Defanged, the bacteria survives. It has edited its invader out of the story. And in that simple act of self-preservation, it gave us a gift we never expected. You drift back upward, rising from the cell with the same lightness you arrived. You are no longer just observing, you are remembering. The DNA you carry is not passive, it is not fixed. It is a manuscript, yes, but now, for the first time in history, we hold a pen, or perhaps a scalpel made of light. With CRISPR, we can change the letters, we can fix mutations, we can delete disease, we can even dream of what else might be written, but not tonight. Tonight is not for dreams of control. Tonight is for reverence, for returning to the quiet knowledge that you are built not of code, but of centuries of memory folded into cells. The scissors have not yet been lifted. The edits have not yet been made. You lie in stillness, knowing that the genome continues, whether touched or untouched, whether read or unread. It curls within you like a sleeping ribbon, breathing, waiting. Hold on, and as your breath deepens into sleep, one last question curls softly at the edges of your thoughts. If the letters of life can be rewritten, what new stories might we gently begin? Beneath everything you see, beneath the forests, cities, oceans, and veins of stone, beneath the pulses of your own blood and the thrum of your thoughts, there lives a layer so old and so quiet that we rarely think of it. Bacterius, more invisible, ancient, have been here since the very beginning. Long before eyes blinked open or lungs filled with air, these single-celled survivors floated through primordial waters, learning the rules of existence. They are not simple. They are not weak. They are, in many ways, the most enduring storytellers life has ever known. And somewhere in the vastness of their invisible world, a war began. Not of violence, but of memory. You begin this chapter adrift in warm, ancient oceans. The sky above is violet, tinged with methane. There are no trees yet, no birds. The earth is young, steaming and restless, but already alive. You drift downward into the shallows, and there they are, bacteria, moving in their simple, purposeful way. They do not have eyes or limbs or minds, but they have something else, awareness. Not the awareness of thought, but the awareness of patterns, of cycles, of change, of danger, because danger, too, is ancient. Viruses stalked these waters even then strange, parasitic slivers of life that could not live on their own, but only by invading. A virus does not knock, it enters, it latches, it injects. Its only goal is to rewrite, to use the cell's machinery to copy itself again and again until the host bursts. The bacteria are small, but they are not helpless. Over countless generations, they did something remarkable. They began to learn. Not in the way you learn with lectures, with words, with deliberate reflection. Their learning was biological, etched into their code with every passing attack. Some bacteria, through luck and time, survived viral invasions, and something extraordinary happened. They kept pieces of the virus inside their DNA, not to copy it, to remember it. These fragments became warnings, filed away like fingerprints in a growing archive. This archive lived in a very specific place in their genome, sure, repeated sequences separated by bits of viral DNA, a pattern, a rhythm, and eventually this became known as CRISPR, clustered, regularly interspaced, short, palindromic repeats. It sounds complex, and it is, but to the bacteria, it is simply survival. When a virus returns, the bacterium consults its archive. It sends a guide strand of RNA shaped to match the viral intruder. This guide leads a protein, a Cas enzyme, to the matching code. And then, with precision sharpened by evolution, it cuts. The virus is disarmed. The memory has served its purpose. You float beside one of these bacteria now. You are small, almost weightless, barely a thought. Inside, you watch the archive being copied. A new piece of virus is recognized, clipped, and filed. A small piece of the enemy turned into protection. You begin to understand how elegant this system is. Not aggressive, but precise. Not chaotic, but curated. The bacteria are not merely reacting. They are documenting. They are building a biological library of what to avoid, of what tried to change them, and of how they survived. In a world without eyes, CRISPR is a way of seeing. In a world without memory, it is a form of recollection. It is both weapon and witness, and perhaps, most beautifully, it is shared. As bacteria divide, they pass on their memories, not through story, but through sequence. Their children carry the same defense, the same archive, the same knowledge. Imagine that for a moment, a lineage, not of spoken tradition, but of molecular editing. A family history built not in words, but in code. Now let your mind rise, slowly. Through epochs, the world changes above you. Oceans shift, plants bloom, animals crawl from the sea. But the bacteria remain, everywhere, still copying, still remembering, still editing. They live in soil, in lakes, in your mouth, in the boiling springs of Iceland, in the cold, dark crust beneath the ocean floor. They endure, they whisper, and in their tiny spirals, CRISPR persists. It isn't until billions of years later that human beings discover this. At first, it seems like just another strange pattern in bacterial DNA, these repeating sequences and odd viral fragments. But then a few curious scientists begin to look closer. In the early 2000s, researchers in Japan and Europe noticed something odd. The viral fragments stored in CRISPR match known phages viruses that attack bacteria. It begins to dawn on them. This is a system, a defense, a memory bank. And then, in 2012, two women, Jennifer Doudna and Emmanuel Charpentier, do something astonishing. They not only understand the CRISPR-Cas system, they reprogram it. They show that the guide RNA can be designed, pointed at any gene in any organism, and the Cas9 enzyme will cut there, with purpose, with specificity, with the same precision bacteria have used for millennia. But now, in human hands, this ancient bacterial weapon becomes something new, a tool, a key, a way to edit the very script of life. You sit with that for a moment. Billions of years ago, a microbe learned to defend itself. Now, that same strategy is the basis for a revolution in genetics, medicine, agriculture, human identity. The quietest beings in the biosphere held, all along, the mechanism for rewriting existence, and they did not keep it hidden. They passed it unopenly in their genomes, waiting for us to see. You feel no arrogance here, only humility. The more we study life, the more we find that the most powerful tools have always been with the smallest creatures. CRISPR is not a weapon we invented. It is a language we finally learned to read, a whisper from bacteria saying, "Here is how we survived." Maybe you will, you rise slowly from the bacterial world, carried by currents of memory and time. You pass through the cells in your own body, where CRISPR systems, introduced by scientists, are beginning to rewrite what once was fixed. You see red blood cells untouched by disease. You see lung tissue free of inherited error. You see seeds sprouting from engineered genomes, thriving in dry soil. These are the ripples of that ancient bacterial memory. These are the echoes of survival, gently guided by new hands. But the story is just beginning. We are infants in the realm of editing. We understand only a fraction of the implications. The bacteria, in their quiet way, remind us, use the scissors with care, use memory with reverence. Do not rush, do not cut without wisdom. Remember that what is edited is still a story, and stories, once changed, cannot always be restored. As your breath deepens and your body settles, the image of that floating bacterium stays with you. Small, silent, powerful. Inside it, the archive grows, one memory at a time, not for conquest, but for continuity. And inside you, too, the memory of this chapter lingers, that sometimes the most profound revolutions begin not with noise, but with pattern, with repetition, with survival, and as the night darkens around you and your eyes grow heavy, a final thought drifts across the surface of your mind like light on still water. If even bacteria could learn to remember, what else might we inherit from those who came before? You find yourself now in a space not grand nor echoing, but small, focused, and filled with the low hum of quiet thought. The laboratory is not a place of spectacle. It is a sanctuary of wonder wrapped in silence. Soft lights glow above polished counters. Glass beakers catch and scatter their shine. There are notes pinned to walls, numbers drifting across computer screens, the soft tap of gloved fingers against pipettes and vials. The smell is sterile, but underneath, you sense something more ancient, the breath of discovery, as familiar as starlight, as persistent as curiosity. It is here, in this humble petri dish, that an ancient story begins to reach human eyes. You float beside a woman bent over a microscope. Her brow is furrowed, not in frustration, but in reverence. She is looking at something so small, so easily missed, yet it holds within it the key to a new age of biology. It began with patterns, repeats, odd sequences inside bacterial genomes. That made no sense, short DNA motifs spaced at regular intervals. She traces them again and again, wondering why they appear not once, but many times, and why, between these palindromic repeats, there are fragments that look familiar, fragments that seem to echo viral sequences. In labs across the world, in Osaka, in Madrid, in Berkeley, and you methods are noticing it too. Different teams, different languages, one shared question: what are these strange, repeated codes doing? In bacterial DNA, it does not scream for attention, it whispers, and only those who lean close hear its message. This isn't the first time life has hidden secrets in plain sight. For years, these repeats were dismissed as genetic noise, bacterial nonsense. But slowly, as if the code itself is growing tired of being ignored, the pieces begin to fall into place. The repeated segments are not random. The interspaced viral fragments are not junk. They are memory encoded and passed down. They are a bacterial immune system, older than any antibody, more precise than any guess. CRISPR is not an invention. It is a revelation. You hover now over a petri dish, warmed gently by the lab's incubator. Inside, bacterial colonies bloom, milky patterns spreading across gelatin-like stars, forming in a newborn galaxy. They are alive, yet they hold still long enough to be studied, watched, understood. The scientists introduce phages, viruses that prey on these bacteria. Most die, but a few survive, and when their genomes are read, something astonishing is found. Pieces of the phage DNA have been added to their CRISPR region. The invader has been filed away. The archive grows, step by careful step, a picture forms. The bacteria are not only storing enemy sequences, they are using them by producing small guide RNAs that match the stored viral code. They lead the Cas9 protein directly to its target, like a map leading a sword to its sheath. A clean cut is made. The virus is neutralized. The bacteria survives. Precision born of pattern. Survival born of memory. The realization stuns. The possibilities begin to unfold in the minds of those watching. What if we could take this system, this natural, ancient form of editing, and guide it ourselves? What if we could design the guide RNA to point not to a virus, but to any gene, any sequence we wish to alter? What if this bacterial defense could become a universal tool for rewriting the language of life? You drift now to another lab. It is 2012. Two women, Jennifer Doudna and Emmanuel Charpentier, stand side by side. Eyes alight with quiet certainty. They are not theorizing in the abstract. They are holding the future in their hands. Together, they refine the CRISPR-Cas9 system. They strip it down, understand its workings, and demonstrate that it can be directed intentionally, cleanly, with purpose to any DNA target. The scissors can cut where we tell them to. The genome, once sacred and untouchable, now has doors, and we are learning to knock. You feel the air shift around you, not with fear, but with immense gentleness. This is not the birth of chaos. It is the soft unfolding of clarity. Science does not always arrive with trumpets. Often it comes like this: a few minds in a quiet room, seeing something everyone else has missed. The implications are vast. What took bacteria billions of years to evolve, humans can now use in hours. The code of crops, of animals, of human cells debatable, not through brute force, but through the precision of molecular memory. Genetic diseases that were once unchangeable begin to shimmer with possibility. Laboratories bloom with research. Clinical trials stir to life. The Petri dish becomes not just a tool for culture, but a canvas for the future. You drift further. Into this space, your mind like fog over warm glass. Everything here is deliberate. The tools are gentle. The tone is reverent. These scientists are not gods, they are gardeners. They know what CRISPR can do, but they also know what it means. That we are no longer simply readers of life's script. We are, in cautious measure, its editors. Still, the lab is not a place of answers. It is a place of questions. How far should we go? How much change is safe? Who decides what deserves editing, and what must be left whole? The Petri dish does not answer. It only reflects the choices placed within it. You step back now, the microscope fading into mist. What remains is a sense of scale, not of size, but of time. Bacteria, silent and unassuming, carried the key, and humans, with all our complexity, had to learn how to listen. The CRISPR system was never invented. It was discovered, like a fossil inside a stone, like a message inside a song you've heard all your life but never understood, as your body softens into stillness and sleep curls at the edges of your thoughts. The image of the lab lingers. The quiet hum, the steady hands, the Petri dish, cradling the earliest forms of tomorrow. You do not need to know every detail. You only need to remember that life holds more memory than we imagine. And when we listen gently enough, it begins to reveal the way forward. And as you sink into sleep, the question that glows gently in the distance is not, can we edit life? It is this: can we carry that responsibility with the same quiet care in which it was found? You are drifting now between strands of DNA like a feather caught in windless air. There is no sound here, no jostle or vibration, only the slow pulse of molecules moving with ancient certainty. You float in the warm stillness of a cell, where life edits itself with hands too small to see and too perfect to need to be seen. This chapter is a soft invitation into the mechanics of CRISPR, not as diagrams or equations, but as choreography, as a delicate molecular ballet. One strand whispers the way, and the other snips cleanly along that whisper's curve. There is no rush, only precision and the immense silence of knowing exactly where you are going. Inside every cell, the genome sleeps. Long threads of DNA lie coiled like unread scrolls, like lullabies tucked beneath your skin. And then something awakens, a guide. RNA is born short, single-stranded, written not to speak aloud, but to point. It is shaped by a memory, crafted from a piece of an invader's code, or, in our hands, designed to match any gene we choose. It is not the cutter, it is the map, the soft guide that tells another molecule where to go and what to do when it gets there. The guide RNA finds its partner, protein, called Cas9. You watch them meet. There's no drama, no spark, just a natural fit, like puzzle pieces that have been waiting for one another since the first breath of Earth. The guide slips into a groove in Cas9, and together they become a seeking machine. Cas9 is no brute. It does not cut without purpose. It listens to the guide. It waits for the match. They travel now through the cell, floating past endless letters in the DNA script. A, T, C, G, again and again. Most of it passes by in silence. Cas9 does not act unless the letters line up, and then it happens, a perfect match. The sequence in the guide RNA aligns with the sequence in the DNA strand. Not similar, identical, a lock and its key. Cas9 pauses, the air stills, and then it cuts. Not wildly, not jaggedly. It makes a double strand break clean, exact. A single snip through the double helix. A pause in the song of life. For the cell, this is no disaster. It is a signal. It begins to respond. It tries to fix the break. And here is where CRISPR shows its quiet power. We can choose what happens next. Sometimes we let the cell repair it imperfectly. A letter or two gets lost, shuffled, miscopied. In doing so, the original gene is disrupted, silenced, made inert. This is called knockout. Other times, we provide a new template alongside the scissors, a rewritten piece of DNA that the cell can use to fix the cut perfectly, but with our version instead. This is no-kin. We do not force it, we invite it. The cell itself does the mending. We only offer a new way to heal. You float beside this moment, the break, the repair, the rewriting, and you do not feel fear. You feel awe. CRISPR does not storm the gates of life. It walks gently through the open door, whispering suggestions. It does not create from nothing. It revises. It refines. It listens. Now your mind drifts across scales. You are no longer in one cell, but in many. In embryos and skin cells, in plant leaves and neuron cultures. In each, the same process unfolds. A guide RNA carries its map. Cas9 follows. The target is found. The cut is made. The cell begins to rebuild. The possibilities are infinite. Edit. Automutation in a child with sickle cell disease. Silence a faulty gene in cancerous cells. Correct a single base pair responsible for blindness. And it all begins with molecules dancing to the rhythm of sequence. You see now how scientists design these guide RNAs. On a screen, they type in the gene they want to target. The software generates a sequencer, simple string of letters. They synthesize it in the lab, load it into the Cas9 protein, and send it into cells. From code to cure, from memory to movement, all in silence. And yet there is something even more remarkable, the simplicity. CRISPR Cas9 is not a sprawling machine. It is a guide and a pair of scissors, nothing more. But that simplicity masks an elegance that took billions of years to perfect. Bacteria built this, nature refined it, and now we, humbled by its precision, try to walk its path without disturbing the soil. You feel the weight of responsibility begin to settle, but not in a heavy way. More like the soft press of gravity that reminds you you're still grounded, still part of this world, even as the science grows bigger than the mind can hold. These are not toys, these are tools. Scissors can trim or tear, maps can lead or mislead. It is not just about where we can cut, but why and when and how much. Still, your breath is steady. This is not a horror story. It is a love letter to what is possible when curiosity listens carefully. CRISPR doesn't promise perfection. It offers precision. It doesn't end suffering in a flash. It gives us a way to begin. But the choice of what to cut, what to keep, what to leave untouched, that belongs to us. As you float back from the molecular ballet, as the scissors close and the map folds gently away, you begin to sense the deeper rhythm beneath it all. Not just the rhythm of enzymes and strands, but the pulse of human longing for healing, for understanding, for control tempered by compassion. CRISPR is not just a technology, it is a moment of reflection made molecular, a pause in the story of biology where we are allowed to thaw just a little. You feel the Cas9 protein fade from view, the guide RNA dissolve like breath in morning light. What remains is silence. The silence of potential, of questions not yet answered, of blueprints waiting to be drawn. And as sleep drapes itself over your shoulders like a soft shawl, one gentle truth remains. When we carry scissors this sharp, our hands must move not quickly but with care, like those who remember that life is not just written, it is lived. You float now through a library not made of pages, but of pulses and patterns, of twists and coils suspended in the invisible softness of a living cell. The genome stretches before you, not as a tangled mess, but as a long, spiraled scroll, one that has never stopped being written. It is ancient and alive, for letters T, C, and G repeating in billions of combinations, each one forming a syllable of your story. And as you hover close, you begin to see what the CRISPR system has offered us. Not a new book, but a way to change the spelling of life, one letter at a time. You are not here to watch sweeping revisions or catastrophic deletions. You are here to witness precision, a single base per two letters bonded in the middle of that helix is enough to decide whether a protein folds correctly, whether a cell knows what to become, whether a body will fight or fall ill. It seems almost too small to matter. But in the language of biology, a single letter can be everything. Take sickle cell disease. One letter out of more than 3 billion is off. A T is replaced by an A, and suddenly the red blood cell's elegant roundness collapses into a crescent. It can't carry oxygen properly. It clogs the body's vessels like gravel in a stream. Generations have suffered for this one tiny slip, but now, using CRISPR, scientists gently correct that. Single typo, the letter is restored, the sentence is healed. The body, once burdened, breathes again. You drift beside this moment, where the tiniest change brings the deepest relief. Not by force, not by chance, but by a whisper of code. The genome listens, it doesn't resist, it only waits for the right signal. And in CRISPR's guidance, it finds the voice it recognizes, one that speaks the same language it always has, just with a different intention. The guide RNA leads Cas9 to the exact sequence. The cut is made. Then the cell, offered a corrected template, builds the strand anew. The new letter slides into place with the smoothness of memory returning home. Nothing extravagant, just one fragment, reformed. The effect, however, is profound. A lifetime of pain begins to unwind. A future once feared begins to unfold. All because a single letter found its way back. You pass now into other cells, other lives. A child born with a rare immune disorder, unable to fight off the smallest infection, is treated with a CRISPR-corrected stem cell. The edit restores a missing function, and suddenly the child's body recognizes danger, produces defenses, begins to grow strong. Elsewhere, in the eye of someone with inherited blindness, a faulty gene is delicately trimmed. Slowly, color returns. Shapes, shadows, light. For the first time, the world isn't something imagined, but seen, and yet this is not science fiction. This is now. These are not dreams. They are trials, studies, treatments. The work is slow, because it must be. Every letter counts. Every correction is a responsibility. CRISPR doesn't overwrite the entire book. It doesn't start over. It turns. The page scans the line and, with astonishing care, adjusts what no other tool could. It is not about invention, but restoration, not replacement, but refinement. And with each edit, we become something more deliberate, not new, but better. Understood, you now see how researchers approach this work. They do not seek to flood the genome with edits. They seek clarity, targeting one gene, one mutation, one condition. They are not scribes in a rush. They are editors with reverence. Each cut is modeled first in silico, tested on simulations, rehearsed before it ever reaches the nucleus of a living cell. CRISPR is powerful not because it is fast, but because it allows for patience. It gives us the ability to wait, to watch, to guide without force. But not all edits are for healing. Some are for understanding. You float into a lab where scientists use CRISPR to silence a gene in a mouse, not to cure, but to observe, to understand. What that gene controls. Without it, does the mouse forget? Does it feel less fear? Does it sleep differently? One by one, the mysteries begin to open. Not because we knock down doors, but because we gently lift the locks. You pause over plants, whose genes have been altered not to make them unnatural, but more resilient. Wheat that can grow without as much water. Rice that resists disease, tomatoes that ripen slower, lasting longer. These are not monstrous creations. They are carefully tended letters, tweaked in a sentence the earth has already begun. We are not rewriting nature. We are learning to read her margins and understanding how much beauty rests in a few quiet edits. And still, the ethical air grows heavier. For every success story, a question lingers. If we can change a letter to cure, can we change it to enhance, to choose eye color, intelligence, height? If editing is easy, does it become casual? These are not answers you can touch yet, but they drift near, like fog across glass. And CRISPR, silent as it is, does not answer. It only opens the page. There is wonder here, and danger too, but wonder leads. The scientists you observe are not reckless. They carry their tools like relics, with sleeves rolled and breath held. They know that behind every letter is a life, and behind every life is a story. You return now to that spiral of DNA, soft, humming, almost musical. You are not outside it. You are inside it. Your own cells are whispering their code even now, maintaining the language they've carried since you were just one fertilized cell. What would it mean to change it? Would it still be you? Would the story hold? These questions do not wake you, they soothe you, because CRISPR is not a bladed, is a brush, and each letter changed is a stroke made by steady, careful hands. The chapter closes, and the genome coils gently back into its resting shape. You feel the presence of possibility, not of conquest, but of communion. The code of life is not sacred because it is unchangeable. It is sacred because it is shared, across species, across time, across breath, and as your body softens into sleep, a single line remains, echoing in your chest like the beat of a lullaby, too quiet to end. We are not rewriting life. We are learning how to read it with care. You step now into a landscape not made of circuits or cells, but of wind moving through wheat, of roots sighing softly beneath the surface, of leaves curling with the first morning light. The air here is warm with soil and memory. There are no machines humming, no sterile counters or glowing monitors. This is the lab of the earth, and in this living green, CRISPR has begun to take root not as disruption, but as quiet dialogue. It enters not to dominate, but to ask, how can we help you thrive? You walk slowly through golden fields of rice, their stalks tall and certain, leaves trembling like breath. The plants here have not been engineered into something alien. They grow as rice always has, whispering in the wind, reaching toward sky and sun. But deep within their cells, a small change sleeps. A gene once vulnerable to a particular fungus has been edited one tiny tweak, enough to let the plant recognize the threat earlier, to resist, to endure. No chemicals were sprayed, no foreign DNA was added, just a letter shifted, a whisper adjusted. You kneel beside a tomato vine, its fruit full and glossy, still clinging to the stem. These tomatoes do not spoil as quickly. Their cell walls remain, strong for longer, their flavor intact. It's not a miracle, it's an edit, a soft rebalancing of a gene that once told them to ripen too fast. Now, the fruit waits, it lasts. And with that, less waste, fewer shipments lost to rot, fewer mouths left hungry. This is not the industrial age of agriculture. It is something gentler, something smaller, yet far-reaching. Scientists are using CRISPR to listen to the plants, to ask them where they struggle, and to offer them tiny tools to face what comes. Drought, pests, flood, nutrient loss, all met with a soft, molecular nudge. A word changed in a sentence the plant has been writing since the first root split. Stone, you move from farm to forest. Here, CRISPR is not helping us grow. Food is helping us remember what was lost. You find yourself beneath the shade of a chestnut tree. Its bark is smooth, its leaves broad and golden in the lowering light. Once, these trees ruled the eastern forests of North America. Then came a blight of fungus, to turned giants to ghosts. But now, scientists have edited a gene to allow resistance, using CRISPR not to invent something new, but to resurrect something forgotten. Nearby, ash trees whisper. They too face a threat, a beetle, emerald and bright, that bores into their bark and leaves them hollow. With CRISPR, a gene may be borrowed or returned, allowing the tree to defend itself again, not by making it unnatural, but by returning to it a forgotten strength. You walk farther still, into colder winds and higher altitudes. Here, in the fragile forests of the future, scientists are editing trees not only for disease resistance, but for carbon capture. A tweak in the pathways that build lignin, the woody backbone of trunks, allows faster growth, stronger structure, more carbon drawn from air and stored in bark and soil. A forest becomes not just a shelter, but a steward. And yet the edit is subtle. You cannot see it in the leaf or trunk, only in time. Still, there are those who hesitate, and you feel their presence too, like a hush on the wind. Is it right to edit a tree, to change a forest's story? CRISPR's hand is small, but its reach is long. The line between help and harm is thin when you work with life, and yet the trees do not resist. They only grow, and perhaps, in their growth, we see our intentions mirrored back. Now you rest beside a plant so small it could be missed among stones. It is a mustard weed, Arabidopsis, model organism, humble but endlessly revealing. In labs across the world, this tiny plant has helped scientists test CRISPR edits before they reach larger species. Every flower it makes, every seed it drops, carries the quiet mark of exploration. Not to design new forms of life, but to understand the old ones better. You remember then how nature edits itself, through mutation, through adaptation, through the quiet filters of time. CRISPR is not so different, only faster, more precise. In this light, it becomes not a disruption, but an acceleration, a way to help evolution do what it always has, just with a gentler hand. And yet, editing the wild must be done with humility. For every gene we understand, 1,000 more remain in mystery. A trait we change in one species may ripple through an ecosystem in ways we cannot predict. The deer that feed on the new crop, the birds that nest in the stronger trees, the insects drawn or repelled. Nothing in nature happens in isolation, so the work goes slowly. Not because CRISPR is weak, but because caution is strength. Trials are long, edits are limited, and always the wild is watched with reverence. Not every tree will be edited, not every seed will be changed. But where life faces extinction, where a blight has already rewritten the forest, CRISPR may offer a footnote, a rescue, a chance to remember how it once felt to thrive. You now drift back across the green world, past rice fields, tomato rows, wheat that bows low, forests that hold breath, and you feel not awe in the power of CRISPR, but in its restraint, in how it listens, in how it asks instead of commands. The edits are not declarations, they are questions. Small molecular questions placed gently into the soil, where time and sunlight will answer. Perhaps that is what makes CRISPR so quietly beautiful. It does not invent meaning, it reveals it. It brings forward what was already written in the seeds and roots and leaves, and simply helps it speak more clearly. And as you begin to soften into the quiet of sleep, one thought settles like pollen on your chest, that we are gardeners, not gods. And even when our tools are molecular, our hearts must remain tender. You drift now through a chamber of light, soft and warm as the inside of a dream. Around you, cells begin to divide, quietly and without ceremony, one becoming two, becoming four. You are watching the earliest flickers of human life. Not a person yet, but a whisper of potential. A hope still curled in possibility. These cells do not know names. They do not yet feel pain or hold memory. They are blank pages, written only with the inherited breath of those who came before. And now, with the guidance of CRISPR, someone has reached gently into that breath and touched the sentence before it was spoken. You are not watching science fiction. You are watching a moment that has already happened. A moment that still echoes through conference halls and quiet hospital rooms, through ethics boards and hushed debates. In 2018, the first reported CRISPR-edited babies were born twin girls, with a small change made before their first cry. A gene was disabled, one associated with vulnerability to HIV. The goal, the researcher said, was to give them protection, not enhancement, not selection, only a kind of molecular shield. And yet, the world trembled not because of what was done, but because of what it meant. You hover now over that moment in time, and it feels not like a triumph, but like a question. The science worked, in part. The gene edit was made. The babies were born. But CRISPR's quiet touch, so precise in a dish, in a leaf, in a mouse, was now brushing across the blueprint of a future person. And suddenly, every cut became more than a correction. It became a choice. You float beside an embryo, only hours old. The CRISPR system has entered the cell. A guide RNA brings its target. Cas9 makes its cut. But in embryos, the repair process is less predictable. Sometimes, only one cell is edited. Other times, a mosaic forms, some cells with the edit, some without. The embryo continues to divide, and with it, uncertainty spreads. The simplicity of the tool meets the complexity of life, and the result is not always harmony. This is the heart of the question. Not can we, but should we? CRISPR offers healing at its most profound depth. Imagine a child spared from a cruel genetic diseaser, single letter corrected before birth, and a lifetime of suffering unwritten. For many, that is not controversial, it is kindness. But the line between healing and enhancement blurs. If we can remove a disease, can we remove weakness? Can we increase strength, intelligence, beauty? And if we can, should we? The air around you thickens, not with dread, but with gravity. This is no longer a conversation about molecules. It is about meaning, about what it means to shape a human before that human can speak, about whether the story of a person should be edited before the story has even begun. You pass now through research labs and policy rooms, through places where silence has given way to debate. International committees meet, scientists draw lines. Many agree we are not ready. Germline editing, the editing of embryos that can pass those changes to future generations, should be paused, restricted, studied. There is too much we do not yet know, too many risks, too many echoes that might reverberate for centuries, and yet the technology moves forward. In animal models, CRISPR has corrected genes for blindness, for heart disease, for muscular disorders. In theory, these same edits could be applied to human embryos someday, with greater precision and safety. The potential is breathtaking. The responsibility immense. You now drift into a different kind of Ruman IVF clinic, where parents look into the future with trembling hope. They want a child, not a perfect child, just a healthy one. They ask, if a gene could be edited to prevent suffering, would that be wrong? If a disease that runs through generations could be stopped, truly stopped before it begins, would it not be cruel not to act? CRISPR stands here like a gentle ghost. It can be silent or it can speak, and when it speaks, it does not shout. It changes one letter. But in that letter, an entire life may unfold differently. You hover near a thought, one not spoken aloud, but felt in the chest. CRISPR is not a tool of control. It is a mirror. What we choose to edit reflects what we value, what we fear, what we believe a human life should be. And that mirror can be both beautiful and blinding. Now you travel back into the embryo. The cells continue to divide. The edit is made. The child is born. The world watches, not with judgment, but with breath held. What will happen next? Will the change remain safe? Will it pass to future children? Will there be unseen consequences? For now, the answers are incomplete. The girls, if reports are true, are healthy. They grow like any other children, and yet they carry in their bodies something no human before them has had, a piece of the future. Written into their past, you begin to drift away from the clinic, the lab, the embryo, and what remains is not fear, but reverence. CRISPR has given us access to the rawest part of life, the part before history, before memory, the part where choice begins. We can enter that space now, but should we? You float, slowly, through questions too soft to answer. What happens when editing becomes available to the wealthy first? When traits begin to separate us not by genetics, but by design? What will happen to the diversity of the human story if some chapters are erased before they are read? But for now, those are whispers. What is real is this: the first children of CRISPR are here, and they remind us not that we are gods, but that we are learning how powerful it is to touch a story before it begins. You are no longer in the embryo. You are in yourself again, in your breath, your skin, your sleep-softened body, and every cell within you still tells your story, written without edits, shaped only by time and inheritance. And somehow, that is beautiful too. As the world slows around you, as the chapter fades into the quiet, one final thought lingers, weightless as light. We are not just learning how to edit life. We are learning what kind of life we want to tell. You are floating now through something not quite physical, yet more intimate than any cell or strand. The space around you pulses with quiet sparks, neural flickers like fireflies in a dark forest. This is the realm of memory, not as you usually think of it, but as it lives inside the folds of your brain. You drift through dendrites and synapses, along electric paths sculpted by repetition and time. And here, in this fragile cathedral of thought, CRISPR has found a place not as a scalpel, but as a kind of lens. You watch as a scientist moves gently beneath a microscope. A mouse breathes slowly nearby, untroubled. The goal is not to harm, but to understand. A single gene one involved in memory consolidation is targeted. The CRISPR-Cas9 system, wrapped in a viral envelope, is delivered with precision to neurons. In the hippocampus, the edit is simple. A gene associated with forming long-term memories is disrupted. What follows is something astonishing. The mouse can still move, eat, explore, but when placed in a space it once feared, it no longer hesitates. The memory, once written, is no longer accessible. Not erased, but unwound. You feel no fear watching this, only a kind of awe. Because memory, it seems, is not mystical, it is molecular. It is built from proteins and signals, circuits and codes. And CRISPR, by gently modifying these codes, allows scientists to ask questions we could only dream of before. What makes a memory last? What causes one to fade? Can trauma be softened? Can forgetting be a form of healing? You drift beside another experiment, this time a different gene. Here, the goal is not to block memory, but to strengthen it. A protein known to stabilize synapses is upregulated using CRISPR activation. The mouse learns faster, retains more, navigates mazes with elegant ease, not because it is smarter, but because the molecular tools for memory are primed. CRISPR does not make it genius. It simply polishes the mirror where memory is reflected. You wonder now, what if we could do this in humans? What if the fog of Alzheimer's could be lifted by editing genes involved in amyloid processing? Already, studies in lab-grown neurons derived from stem cells of Alzheimer's patients have used CRISPR to correct mutations to reduce the buildup of toxic plaques. The early signs are hopeful. Not cures, but clarities. Tiny lights turning back on in the darkening brain. And yet, memory is more than just storage. It is identity. It is how you know yourself across time. You remember your first breath of winter, air, the sound of a name spoken with love, the ache of a goodbye that never fully left. To change memory, even to heal it, is to touch the very core of being. CRISPR does not yet reach that deep in humans, but the door, once locked, is now cracked open. You drift now through research on fear. In mice conditioned to associate a tone with a mild shock, CRISPR is used to silence the gene that strengthens that fearful memory. The next time the tone plays, the mouse does not freeze. The memory is uncoupled from emotion. It's not gone, exactly, but softened. What would it mean if this were possible in people with PTSD? Could CRISPR help uncouple trauma from the body's deep alarms? Could nightmares be softened at their roots? The science remains young, but it whispers of what may come. CRISPR could help us not just remember, but forget with intention, with care. To trim the thorns without cutting the rose. You are still floating inside the brain now, watching signals dance. You realize memory is not one thing. It is layers, short-term, long-term, emotional, procedural. Each lives in different corridors of the brain, and CRISPR offers a key to each door knot, a universal answer, but a specific invitation. Scientists now map genes involved in memory extinction, in habit formation, in the encoding of joy. Each gene a syllable in the language of recall. Each edit a possible rewrite of how we carry the past. But here, the ethical winds begin to stir. If we can change how people remember, what becomes of consent? What becomes of truth? Could memory be altered not to heal, but to influence? To rewrite not just a genome, but a narrative? For now, those fears remain future tense. The present is quieter, focused on healing, on learning, on using CRISPR not to change who we are, but to understand how we become. You drift through a classroom, now not one with desks, but with glowing neurons. Researchers use CRISPR to tag genes activated during learning. They follow the traces left behind as memories form. They can see the moment when new information takes hold, when a lesson becomes a memory. This is not magic. This is the biology of becoming. You pass near a study using CRISPR to edit genes involved in neurodevelopmental disorders, Fragile X, Rett syndrome, conditions where memory and cognition are affected from birth. Tiny edits lead to improvements. In mouse models, tiny steps, perhaps toward therapies for children who struggle to hold on to the world as it moves by. The mind, you see, is not separate from the body. It is built from cells, just like skin and bone. And if CRISPR can help those cells fire more clearly, more gently, then maybe the mind too can be a place where healing begins. You are nearly asleep now, drifting slowly through a synapse, glowing like a candle in a cave. Memory, you realize, is not only about what we keep, it is about what we let go, about which stories stay and which ones dissolve like breath on glass. CRISPR offers no answers to the soul. It does not define who you are, but it does give you a languagey way to ask how memory is made, how it is held, and how it might someday be healed. And as you drift further into sleep, the final echo of this chapter lingers like a lullaby of light. Perhaps memory is not fixed. Perhaps the self is a story still gently being told. You begin in blood-warm, red, eternal, the river that carries breath, memory, immunity, and ancestry. Each drop teems with cells that know who you are, and some of those cells, through no fault of their own, carry a twist. A mutation passed from parent to child. A single misspelling in the book of life that becomes pain, illness, fragility. For generations, there was no way to correct this. You could treat the symptoms, you could wait, you could hope, but now a quiet revolution has begun. It doesn't shout, it edits. You drift beside a young woman. Her blood contains cells shaped not in perfect rounds, but in crescents. Sickle cell disease, a disorder not of character, but of code. Her red blood cells cannot carry oxygen well. They get stuck, tangled, in vessels. The pain comes suddenly, fiercely, like fire in the bones. But this woman has agreed to something new. Her stem cells are collected. In a lab, CRISPR is used to turn back on a gene from early in life, fetal hemoglobin gene, usually silent. With this gene awake, her body can produce a different form of hemoglobin, one that works, one that heals. The edited stem cells are infused back into her blood, and over time they grow, multiply, spread. The crescent cells become fewer. The pain retreats. Not cured entirely, not yet, but rewritten from the inside out. And you feel the shift. Illness no longer a sentence, but a draft. Now you follow another patient, a child, losing their vision to Leber's congenital amaurosis, a rare inherited form of blindness. In the past, this was a story that could only end one way, but now doctors deliver CRISPR directly into the eye. The edit is made inside the living tissue. No removal, no dish, no long delays. A faulty gene is repaired. Slowly, softly, light returns to the child's world. Shapes appear. Shadows fade. The child points at colors they've never named before. You drift through hospital halls filled with possibility. CRISPR trials are underway for diseases once thought untouchable. Beta thalassemia, Duchenne muscular dystrophy, Huntington's. Conditions with genetic roots now visible, now reachable. The edits are cautious, precise. No sweeping changes, just a letter here, a silencing there. Enough to shift suffering back toward balance. You enter a room where cancer is being met not just with chemotherapy, but with a different kind of warrior. Immune cells taken from the patient are edited using CRISPR trained like watchful sentinels to seek and destroy tumors more precisely. These modified cells are returned to the body where they hunt, where they learn, where they heal. This is not science fiction. This is science now. And yet, each cure begins with a story. Each patient is a chapter in the book we are still learning. How to write, you drift into the liver, where cholesterol is managed, not with daily pills, but with a single CRISPR edit to a gene called PCSK9. One change, one injection, and suddenly the body regulates cholesterol more naturally. The heart beats longer, the arteries stay clearer. The edit stays for life, but not in the way that frightens, in the way that frees. CRISPR is also used to fight infection. HIV, hidden in the genome. Scientists are learning how to use the tool not just to cut, but to seek out viral DNA and disarm it, to delete the hiding places of a virus that has shaped generations of fear. And in other labs, rare diseases are confronted one by one, with each a handful of patients, with each a quiet miracle. A child with Tay-Sachs who might live longer. A boy with SID, once bubble-wrapped in sterile rooms, now playing in fields. These are not mass-market cures. They are personal, precise. Poetic, you begin to see it. Medicine is no longer only reactive. It is becoming proactive. Not just treating what has happened, but rewriting what might. And yet, with this power comes a softer question. What does it mean to cure? If a gene is corrected, is the person new or simply more themselves? The goal is never perfection. It is balance. It is peace in the body. CRISPR cannot edit the soul, but it can smooth the terrain on which the soul travels. You float nearer, cell in the bone marrow. It carries a tiny message of new instruction. This edit, if it works, will pass to every blood cell born from it, a molecular legacy of healing. And so, the body heals itself, not with outside drugs, but from within. But not every story is simple. Some trials do not succeed. Some edits have side effects. Some corrections carry unknown risks. This is why the work is slow, ethical, reversible. When possible, scientists walk with reverence. They know the genome is not a machine, it is a story written over billions of years. Edits must be whispers, not declarations. CRISPR is still young, but its voice is already reshaping medicine. As a roar, but as a breath. A letter changed, and a life changed. A small silence lifted from the body, and still there is caution. Who gets access to these cures? Who can afford them? Will healing belong to all, or only to the few? These questions hover like mist. May bring cures, but it also reveals systems. And systems, like genomes, must be rewritten with care. You return now to the patient. They rest quietly. A nurse checks vitals. A family waits with hope. The edit has been made. And in that patience, blood, something old has been rewritten. The disease, once inevitable, is now in question. The future, once feared, is now unfolding. As you drift farther into your own stillness, the body begins to feel more like a story you are part of, not bound to, but partnered with. Your blood, your breath, your cells still dancing in rhythms, older than memory. And now, for the first time, perhaps a little freer, CRISPR is not a magic wand, it is a pen. And medicine is learning to write again, not with urgency, but with gentleness, not to rewrite who we are, but to give the body back its voice. And as sleep takes you deeper, the final words of this chapter move like a pulse beneath the surface. Healing is no longer only what we do, is what we can now become. You are no longer inside a cell. You are drifting through something larger and quieter, a space shaped by breath, thought, and memory. A space where people speak carefully because the words they choose echo into lifetimes. This is not a lab, but a meeting hall, a hospital room, a dinner table. This is where ethics lives not in rules alone, but in the soft weight of choice. You begin with a whisper, one that came long before CRISPR had a name. It is the whisper of power, of possibility. Every new technology brings with it a question. Who decides how it should be used? And beneath that, a deeper question still. Who is it for? CRISPR is no longer only a tool for researchers. It has entered our stories. It appears in news headlines, in policy papers, in films and novels, and hushed conversations between hopeful parents. It promises the ability to heal, but it also opens a door we have never walked through before. The power to shape life, not after it begins, but before. And the world has paused here, not with rejection, but with breath held. You float through an international summit. Scientists, ethicists, doctors, patients, activists all gathered to speak of boundaries. The consensus is cautious. Germline editing, the altering of genes in embryos that will pass those changes to future generations, is not ready. The science is still uncertain. The social consequences, vast. Some nations ban it outright, others regulate it strictly. Still others remain silent, watching. The debate is not about fear, it is about responsibility. Because when you can change a letter in the human genome, you are no longer only treating illness. You are shaping lineage. You are editing not just a person, but a branch of the human tree. You pass through questions too large to answer. What defines disease? Is deafness a disability to be corrected, or a culture to be respected? Is short stature a medical issue, or a human variation? When does correction become erasure? When does help become harm? You drift beside a parent holding a newborn child. The child has a rare genetic condition. The parent loves them deeply, but wonders if CRISPR had been available earlier, could the child have been spared this pain? Is choosing to edit a child's genome an act of love or an act of control? In another room, a patient with Down syndrome speaks at a forum. They do not want to be erased. They do not want to be cured of who they are. They want to be seen, valued, supported. CRISPR, they say, should not be used to create a world where only some kinds of life are welcome. You hover over a whiteboard in a university lab, where students debate designer babies. They imagine enhanced trait sight, intelligence, athleticism. But each enhancement, they realize, creates new inequalities. Who will afford these edits? Who will access them first? Will they divide humanity into castes not of wealth alone, but of biology? Already, inequality exists in access to basic healthcare. Will CRISPR widen this divide? Or can it be harnessed to reduce it? Can we imagine a future where the benefits of gene editing are shared, not hoarded? You pass near an ethics review board reading through a clinical trial proposal. Every word is measured. Every edit considered not only for safety, but for meaning. They are not just asking, will this work? They are asking, who might be hurt by this? Whose voice is missing from this decision? For too long, science has moved. forward with only certain voices at the table. CRISPR forces a new kind of listening. The future it opens will not be shaped only by biologists, but by poets, philosophers, parents, children, by those who have been excluded, by those who carry the weight of history in their blood. You drift through a library, now its shelves filled not with genes, but with stories, myths of creation, legends of gods shaping people from clay or stars. CRISPR is not divine, but it echoes those myths. It invites us to shape what we are. And so, we must ask, who gets to write the next chapter of human evolution? Consent becomes a question, here not for patience alone, but for generations unborn. How do we speak for those who cannot yet speak? How do we ensure that the choices made now will not limit the choices of those to come? You feel the tension between healing and hubris, between care and control. CRISPR is not inherently good or bad. It is a mirror. It reflects what we value. It amplifies what we believe. And like all mirrors, it can distort if we do not look with care. Some propose moratoriums. Others propose international guidelines. But the deeper work is not in regulation. Alone it is in culture. In creating a world where the decision to edit a genome is never made lightly, where humility walks beside knowledge. You drift beside an indigenous scientist who reminds her, colleagues, that many cultures have long held deep respect for ancestry, for the stories written into blood. She does not reject CRISPR. She simply asks that we proceed with reverence, that we remember our place not as masters of life, but as stewards. And so you return to the central question, who gets to rewrite life? Not just those with tools, but those with stories, with memories, with care. You return now to your own breath, your own body. You may never edit a genome, but you live in a world shaped by this choice, and you have a voice. You have the ability to ask, to wonder, to hold space for complexity. Ethics is not about fear. It is about love. It is about saying, this matters. Let us not rush. Let us not forget the ones left out of the conversation. Let us not trade mystery for convenience. Let us not edit away what it means to be human. And as you drift deeper into silence, the final thought of this chapter stays with you, not an answer, but a question with open hands. In the age of rewriting life, what will we choose to remember? You. Begin your drift at twilight, the time when wild things stir. A field stretches before you, warm with dusk and folded in tall grasses. From beneath the soil to the arch of the clouds, life speaks in patterns. DNA lives here too, in roots and wings, in feathers and bark, in silent codes passed between pollinators and bloom. And now, for the first time, CRISPR is entering the wild, not as an invader, not as a cure, but as a question. You float near a quiet hut in West Africa. Inside, scientists sit beside villagers, not apart, but together. They speak of a small insect with a vast reach, the mosquito. Not all are dangerous, but some carry malaria, which has shaped entire generations in fever and grief. CRISPR is being used to edit a gene in Anopheles mosquitosa, fertility gene. When these edited males mate, the trait spreads, and the mosquito population declines. You drift across wetlands where these CRISPR-modified mosquitoes have been tested. The goal is not extermination, but balance, to relieve the burden of disease without harming the ecosystem. But the question ripples outward. What happens when one species fades? What other lives depend on it? What songs are silenced when we strike a single note from nature's chord? In another part of the world, corals are dying. The ocean warms, and the reefs bleach like forgotten bones beneath the sea. But in a marine lab, scientists are editing corals with CRISPR, modifying their genes to make them more resilient to heat. These changes are not dramatic, just enough to help them hold on, to survive what should have been impossible. They are not building a new reef. They are trying to keep one alive. You drift gently through a forest in North America where a tree, once known as the American chestnut, once reigned tall, proud, and then gone, erased by a fungal blight. But now, a CRISPR edit may bring it back. A single gene inserted to resist the blight, and the tree grows again, quiet and strong. The forest may yet remember itself. CRISPR is not only editing species, it is shaping futures. Gene drives tools that ensure an edited gene spreads quickly through a population are being considered to fight invasive species, to protect endangered ones, to rebalance islands where non-native animals have tipped the scales. But each edit, each release, is a story written not only in science, but in soil and air. Who decides what nature should look like? Who decides what to protect and what to change? You drift beside an indigenous elder watching a river. They do not fear the science, but they ask for a slower pace, for inclusion, for respect. CRISPR cannot replace millennia of ecological knowledge. It must move in conversation, not command. In Australia, scientists use CRISPR to edit cane toads, an invasive species that has decimated native life. In labs, they study how to turn fertility against the invader, how to gently decrease the population over time, not with poison, not with force, but with code. Still, the question stirs again. Are we restoring nature or reshaping it in our image? In the Arctic, scientists speak of bringing back the woolly mammoth not as a fantasy, but as an ecological tool, to rewild the tundra, to slow permafrost melt. This would not be resurrection, exactly. It would be a hybrid mammoth genes edited into elephant cells, an echo, not a return. The line between conservation and creation begins to blur, and so you begin to see the contours of a new kind of relationship. The earth, long observed and mourned, is now also being edited. Not from afar, but within. But every solution brings a new question. Could edited species outcompete others? Could gene drives cross borders, spreading where they weren't meant to go? Could one edit shift entire ecosystems? CRISPR has no intent. It only obeys. But we must decide how and why and when it is used. In nature, balance is rarely simple. Every action echoes. Every silence carries weight. You drift now through a cave where bats sleep, their wings folded. Like old paper, scientists study how diseases like white nose syndrome ravage these populations. Could CRISPR help? Could resistance be written into the next generation of pups? In a desert, a cactus glows under the moon. Its seeds have been stored. In vaults, its genome sequenced. A CRISPR edit could make it more drought-resistant, more able to survive a changing climate. But should we? Should every species be made stronger, or should some be allowed to fade, as they always have? You begin to sense that CRISPR, in nature's hands, is not a scalpel but a seed. It must be planted with care. It grows into something. Larger than control, it grows into responsibility. The earth does not ask for perfection. it asks for harmony. And if CRISPR is to be used here, it must be tuned like a flute, each note carefully chosen, each silence honored. You pass now by. Children planting CRISPR-edited rice. The crops resist drought, pests, disease. The yield is higher. The families eat. The stomach is full. In this moment, CRISPR is not theory, it is life. But elsewhere, monocultures rise. Corporate seeds dominate. Intellectual property claims swirl around ancient grains. Who owns an edit? Who profits from the breath of the wild? You return to a field where a butterfly lands on a blade of grass. It has not been edited. It has not been touched. And still, it is changing. The earth has always evolved, slowly, softly. We are only now beginning to understand its pace. CRISPR moves faster. But maybe, if we learn to listen, it can move wisely. As you lie now beneath stars that once seemed eternal, you realize the wild is no longer only out there. It is within us, around us, entangled with every choice we make. CRISPR does not divide nature and humanity. It reveals that they were never separate. And as sleep folds over you like a leaf over dew, the final echo of this chapter lingers like wind in the trees. We can edit the wild, but can we learn to walk gently enough to deserve it? You are floating now, not through a lab or a forest, but through time itself. It is unmarked, unwritten. A soft, silver fog stretches ahead, and the footprints you leave behind are made not of steps, but of choices. This is the future. Not a prediction, not a prophecy, but a becoming. And CRISPR, quiet and sharp as a feather's edge, has etched itself into the thread of what comes next. You drift beside a cell knot, a natural one, but a designed one. Not to mimic life, but to expand it. Synthetic biology is CRISPR's next realm, not just editing genes, but building them. Designing entire pathways, instructions, systems, life as legible as a blueprint. Already, bacteria have been programmed to sense toxins in water, to glow when danger is near, to build tiny structures like microscopic architects. These are not miracles. They are edits, layered upon edits, shaped into function. You float through a chamber where gene circuits hum like small engines inside living cells. Scientists are creating biological computers where DNA carries logic, switches, memory. Cells that can decide when to act, when to rest. A medicine that knows when it's needed and turns itself off when it's not. You're no longer looking at a tool. You're looking at a new language, biology as code, the body as canvas, and then you drift into healing that once lived only in hope. Universal cell therapy, immune cells edited to fit any patient without rejection. Organs grown in animals with human-compatible genes. CRISP are used not just to correct errors, but to improve resilience, to prevent disease before it begins. Not enhancement for power, but enhancement for balance, for fairness, for life in labs. Scientists imagine using CRISPR to slow aging, not to make the body immortal, but to extend health, to give more years of vitality. Mice with edited genes live longer. Cells repair themselves more gracefully. Age becomes not a wall, but a bend. Still life, still end. But softer. You pass through a classroom where children are learning to read their own genome, not with fear, but with wonder. They understand their ancestry, their risks, their strengths. They make decisions informed by their blueprint, not defined by it. Genetic literacy becomes a birthright, not a luxury, And yet, with each step, the question deepens. What does it mean to be human in a world where we write ourselves? A woman chooses to edit her embryos, genomini to perfect, but to remove the shadow of a fatal disease. She doesn't do it for beauty. She does it so her child will not suffer as she did. Still, the edit passes on. Her child will pass it, too. The line of descent has shifted forever. Not better, not worse, just changed. You drift beside an artist painting portraits of DNA, each one different, each one beautiful. Not for sale, not for science, just for remembering. In a world of programmable life, the need for story deepens, for soul, for imperfection, for scars that remind us we were here. A philosopher gives a lecture on post-genomic identity. Who are you, she asks, when your body is editable? Are you still bound by biology, or are you now a co-author of your own becoming? Elsewhere, communities gather to decide collectively what kind of edits they will allow. Some say yes to prevention, others to repair only, others say none at all. Each choice is rooted not in science alone, but in culture, faith, memory. The future, you realize, is plural. Not a single story, but a library that you pass through a cryobank holding extinct species, DNA preserved like pressed flowers. Scientists discuss whether to bring them back, to restore lost balance. CRISPR could do it. The woolly mammoth, the passenger, pigeon, the dodo. But would they belong? Could we restore the habitat too? Could we restore the time? You drift past Mars, its red soil barren, dreaming of green. If we ever settle beyond Earth, it will not be as we are. We will bring edited bacteria, plants, maybe even edited selves. CRISPR will shape not only survival, but adaptation. The genome will become a traveler's tool, a passport between worlds. And you wonder, is this the end of evolution, or its next beginning? Already, there are voices calling for restraint, others calling for acceleration. Some see salvation, others see hubris, but CRISPR does not pick sides. It is a pen. It writes what we tell it to write. You hover now at the edge of something vast. A child is born. Their genome was edited to repair a fatal disorder. They will live. They will laugh. They will carry the edit, but their path is still theirs. Didn't write their choices, only removed the fog from the path. You begin to see that becoming is not about control. It is about care. We will not perfect ourselves. We will not erase suffering entirely, but we may rewrite some of it. And in that small mercy, a gentler future may take root. You return to your breath now. You are the same and not the same. You carry stories in your cells, some old, some newer. You are both author and archive. CRISPR will not decide the future. We will dot, and as sleep moves gently across the bridge of your brow. The last line of this story breathes itself into your chest like wind over a page. The code may change, but the question will always remain: what will we become now that we can? And now you return, slowly, softly, from that far horizon, from neural networks that bloom in silence, from machines that learn not with eyes, but with patterns, from minds built not by nature, but by design. You have walked through code, and consciousness, intelligence, and ethics, futures imagined and futures unfolding. The questions remain not loud, but deep. Can machines truly understand? Can they feel? Can they teach themselves to care, or only to compute? We do not know, not yet, but we are listening, We are learning, too. And in the quiet between each keystroke, each line of logic, a new kind of story begins. You are still human, still flesh, still fragile, but the world is different now. It breathes in zeros and ones. It thinks in circuits. It remembers more than we ever could. And so you lie back, wrapped not just in blankets, but in the weightless wonder of what is becoming. Sleep well, traveler, for the machines may be learning, but you too are evolving. Good night.